Hi folks, today in lecture 3 we're going to talk about Mendelian inheritance, and before I get started with that I want to just cover some basic definitions that you probably are already familiar with. But just to make sure we're all on the same page, we talk a lot about traits, phenotypes, and characteristics in genetics, and I just want to clarify what all of that means. Traits and characteristics are largely the same thing. Phenotypes are different. So phenotypes are variations of traits. So for example, Mendel examined a number of different traits in pea plants, including seed color, seed shape, coat color of the seeds, the pod shape, pod color, etc. We can also think of hair color, eye color, height. Those are all traits or characteristics. Phenotypes are variations along those traits or characteristics. So for example, yellow or green color seeds, that's a phenotype. The seed color itself is the trait. So for hair color is your trait, your phenotype could be brown, blonde, red, dirty blonde, whatever. Okay. Here is just a table that summarizes some basic definitions that we're going to use in genetics. This table is also available in your book. It's also now available on the slide that will be available on Blackboard. So one other basic of genetics is understanding what makes a good genetic model organism. And this is, again, probably going to be review. You probably already know that good model organisms are easy to rear in the lab. They have large family sizes. They have short generation times. But two other characteristics that are important in good genetic models include we need to be able to breed them easily. And so if you have all of these other characteristics that make good models, including large family sizes, if we can't get two organisms to come together and breed, it doesn't matter how many offspring they have because we can't make it happen intentionally in the lab. Another characteristic that's really good for a genetic model is to have mutants that are readily available. And this becomes a resource for genetic research because if we have and when I say mutants, I mean individuals that are variable that are also homozygous for that variation, such that you can produce a strain of organisms that all have that same phenotype. That's what I mean by a mutant. And Mendel actually had all of these traits, all of these characteristics of good model organisms were present in pea plants. They were easy to rear in a greenhouse. He was able to cross them and self them very easily. They produce lots of seeds. They have short generation times, and he was able to get a hold of different mutations for these pea plants. So plants that consistently produced yellow wrinkled seeds, or plants that consistently produced, short plants versus tall plants. One last genetic characteristic of a good genetic model organism that I would like to talk about that is not really mentioned in your book is the availability of genetic resources. Now, these days, that's incredibly important, and more genomes are being sequenced all the time, and the availability of a sequenced genome is especially important, as are the ability to manipulate genes within that organism. I also want to talk about some terminology around that we use when we refer to genetic crosses. The parental generation is referred to as the P generation. That's the generation where we start out with. In this case, we've got two homozygous individuals that we are crossing. Okay. The next generation, their progeny, is the F1, or first filial generation. In this case, they are all 100% hybrid individuals. Our second filial generation, or F2, is what comes when you cross the F1 generation, and so on and so forth to F3. Again, these are hybrids. This, when you cross the F1, if they are both hybrid individuals, this is a monohybrid cross. This is a cross between individuals that are heterozygous at a single gene or locus, which is where the mono comes in. Dihybrid cross, if you recall, occurs when you are crossing two individuals who are heterozygous for two loci. So here we have round versus wrinkled, and you could have round and yellow versus wrinkled and green, for example. If you were to cross the F1 individual with one of the parents, with the one of the parental strains, that's a back cross. And this particularly applies to plants. If you are crossing two individuals, that's called cross-fertilization. In this case, what Mendel did is he selfed the F1 individual. So he essentially bred the F1 with itself, crossed pollen and ova with from the same plant, and this is self-fertilization. All right, just to review Mendel's famous cross, crossed again homozygous individuals for two different phenotypes of the same trait, produced individuals. He found they were 100% round, and he selfed these individuals and found that the wrinkled, one of the parental phenotypes in the parental generation, came back in the F2 generation. So he started with what we call pure breeding lines that are, again, homozygous. He found that there is no, in this first cross, he found that there's actually no blending inheritance. And this was a common hypothesis that was around at that time, where the idea being that if you cross two unlike individuals, their progeny will be some intermediate between the two. He discovered that in this case, blending inheritance is not what's happening. And here we have the F2 generation. The F2 generation is where the F2 is what we calculate when we're using the Punnett square. So from this cross and a series of similar crosses, Mendel concluded that each parent contributed some genetic factor, and that they were transmitted ultimately to the F2 generation because these guys showed up. It was hidden in this generation, originated in this generation, and then showed up again in this generation. And so he concluded that there's some genetic factor, which we now know to be alleles, that are transmitted through multiple generations. Based on these experiments, Mendel came up with his principle of segregation. And what the aspects of the principle of segregation include the idea that some hereditary factor produces a trait. Each individual carries two forms of the gene or hereditary factor. These are alleles. And one allele is dominant over the other, such that when they are present in the heterozygote, only one allele, only one phenotype is expressed. During meiosis, alleles segregate with equal probability into gametes, and at fertilization, gametes fuse with equal probabilities. So let's pause for a minute and focus on this one. During meiosis, alleles segregate with equal probability into gametes. This is what we covered last time in lecture. And let's think for a second about what stage of meiosis in particular does this aspect of the principle of segregation refer to. So I want you to take a moment here to maybe pause the video and think about it for a minute. Maybe go back to your notes and think about, okay, where in meiosis are we getting independent segregation? And the answer is during metaphase 1. When chromosomes are lining up on the metaphase plate, they're doing so randomly. Just go back to this slide from the previous lecture where this just shows 4 different ways in which 3 different chromosomes of a diploid cell can line up randomly along the metaphase plate. And so you could get alignment like this, you can get alignment like this, etc. Completely random. And that was another aspect of Mendel's principle of segregation. And so you end up with these types of possible scenarios. And so recall that this is metaphase 1, and that in metaphase 2 is really just splitting up the sister chromatids. Okay, so the real variation comes from metaphase 1. All right, so back to today's lecture. It is important to recognize that independent segregation of alleles is true whether or not you are crossing over. So for example, if you get crossing over, you get, if you have no crossing over, you're going to end up with this possible scenario where you have a big R, little r, little r. If you do have crossing over, you still have the same scenario where you have a big R, little r, little r. Now, what independent segregation is talking about is it's talking about alleles segregating independently on different chromosomes. And so whether or not crossing over occurs, crossing over is really mixing alleles up within the same chromosome. It doesn't have to do with different chromosomes. And we'll kind of continue to talk about what's happening on different chromosomes versus what's happening on the same chromosome as we kind of get into starting to think about multiple genes being involved in creating a phenotype, and then we start talking about genetic linkage. But for now, let's just focus on the fact that different chromosomes are segregating into gametes completely randomly and independently. So just to kind of carry this further, the no crossing over still gets you these gametes, 2 R's and 2 big R's, 2 little r's. With crossing over, you still get these gametes, 2 big R's, 2 little r's. So Mendel did not know what the factor was that was being inherited that was associated with phenotypes, but around the same time that Mendel was working, Walter Sutton was studying spermatogenesis in grasshoppers, and he was coming up with chromosomes, or he was discovering chromosomes, and this was around the time that actually that Mendel's work was being rediscovered. So this was around the turn of the century, in the early 1900s. And he was concluding that chromosomes were actually the basis of Mendelian inheritance, based on patterns of inheritance in different grasshoppers. And Theodore Bovary came up with a similar theory around the same time, studying sea urchins, and so they're both credited with coming up with this Bovary-Sutton chromosome theory. That you should definitely check out the supplemental video on dominance and recessiveness. At this point, it's not exactly supplemental. I mean, it's supplemental to this lecture, but it's really, really critical, actually, that you watch that video, because dominance and recessiveness is a point of confusion for many, many, many people, and I kind of want to make it absolutely clear what I mean by it. So please go see that supplemental video, and I will expect you to have watched it and to have a really good understanding of what I mean when I say dominance and recessiveness. Okay, so the Mendelian principle of segregation lets us use basic rules of probability to predict phenotypic ratios. So the first rule of probability that I'm going to talk about is the addition rule. And this is the probability that some event or some other event is equal to the sum of their individual probabilities. So if we're expressing probability P of 2 separate events, X or Y, the probability of one of those happening, X or Y happening, is the probability of x plus the probability of y. The multiplication rule predicts the probability of the simultaneous events x and y, and that is the product of their individual probabilities. So, for example, if we want to calculate the probability of, getting green eyes and blonde hair, we would calculate calculate that by multiplying the probability of getting green eyes times the probability of getting blonde hair. So I just want to give you a few practice questions so you can pause the video and kind of try them on your own. And I want you to calculate the probability of flipping 2 heads in a row first. So I'll just pause it for a minute. The way to think about this is to think about it in terms of flipping 2 heads in a row. You're flipping one head and another head, and so you would calculate that by the probability of flipping a head, which is 0.5, times the probability of flipping a head, which is another 0.5, and that is 0.25. So now, what's the probability of flipping heads or tails in a single flip? And that is the probability of flipping heads plus the probability of flipping tails, which both of those are 0.5, so we end up with a probability of 1. And if you think about a coin toss, if you're going to flip a coin, you are going to get either heads or tails guaranteed. So the probability of getting heads or tails is 100%. Now, what's the probability of flipping a coin twice and getting a head and a tail? And this is a little bit trickier. So pause the video and try to work it out for yourself. So to do this, think about it. This is tricky, and the reason I'm giving you this is because it relates to the probability of getting a heterozygote. So because you're flipping a coin twice, you're getting 2 alleles, you're getting a head and a tail, a big A and a little a. And so the probability of getting a head is 0.5, and the probability of getting a tail is 0.5. So that's 0.125. But there are 2 ways of getting a head and a tail twice, or flipping a coin twice and getting a head and a tail. You can get a head and then a tail, or you can get a tail and then a head. So you're actually looking at 2 separate probabilities. You're looking at the probability. You're looking at the probability of getting a head then a tail, or getting a tail then a head. And so the probability of getting a head and a tail is 0.125. The probability of getting a tail then a head is also 0.125. Getting one or the other, you add them, and that gives you 0.25. Oh, sorry. The probability of getting a head and a tail is 0.25. The probability of getting a tail and a head is 0.25. You add those and you get 0.5. And this is the probability of getting a heterozygote as well in a monohybrid cross. So to look at this, to put these probabilities into the context of the Punnett square, what we have here is what we actually call a test cross. This is a cross between a heterozygous individual, big T and little t in this case, and a homozygous recessive individual, little t. We can calculate the probability of getting a big T individual from this cross by using the probability laws. So the rules of probability. So the probability of getting a big T from this individual is 50%, half, 1/2, 50%. And then we can multiply that by the probability of getting a big T from this individual, which in this case is 0 because it doesn't have a big T. And so the probability of getting a big T individual is 0 from this cross. The probability of getting a big T little t is 1/2 times 1. because we have 100% probability of inheriting a little t from this individual, which gives us 1/2 times 1 is 1/2. And so what we expect 1/2 of our progeny to be tall, big T, little t. Finally, we can calculate the probability of a little t, in which case here our probability of inheriting a little t is 1/2 here. And again, times the probability of getting a little t here, which is 1. So again, 1/2 times 1 is 1/2. So we're expecting half to be big T, little t tall, and half to be big T, little t short. Try that exercise with other crosses in a Punnett square. You can do a monohybrid cross, you can do, anything that you want. So check it out and just convince yourself that the rules of probability are how we get the ratios that we get from a Punnett square. Ultimately, I want you to be able to not actually draw out the Punnett square. I want you to be able to look at a cross and tell me exactly how many of any one genotype to expect from that cross using the rules of probability. So we've been talking about the principle of segregation, and this applies to alleles at one locus. This tells you that you're just as likely to get a big R from a heterozygote parent as you are a little r from a heterozygote parent. So we're only talking about the R locus when we're talking about independent segregation. Mendel's principle of independent assortment extends that principle to segregation, to talk about segregation of alleles at multiple loci. So before we were only talking about a single locus, here we're talking about multiple loci. So in this case, we've got our R gene and our Y gene that are in different chromosomes. And so principle of independent assortment says that we are equally likely in our gametes. Here's our heterozygous individual. Our heterozygote is making gametes. In the heterozygote, we're just as likely to get a big R, big Y in a single cell as a little r, little y, as a big R, little y, and a little r, big Y. Okay, so all of these are equally probable. It's just extending the principle of segregation to multiple loci. In the context of meiosis, this means that alleles at different loci are segregating with equal probability into gametes. We can again use the principles of, or the rules of probability to calculate the expected genotypes from a certain cross based on the individual probabilities. So what do I mean by that? The first thing that you want to do is calculate the expected outcomes for each phenotype separately. So let's say here's our cross, a dihybrid individual crossed with a recessive heterozygote, or I'm sorry, recessive homozygote. So this is also a test cross because the test cross that we saw previously was a hybrid individual, a heterozygote crossed with a recessive homozygote. Here it's just two loci. We have an individual who is a heterozygote at two loci crossed with a individual who is recessive homozygous at two loci, at those same loci. And what we want to do is calculate the expected outcomes for this cross independently. So the big R, little r crossed with the little r is going to give you 1/2 big R, little r, and 1/2 little r. And you can calculate that based on the probability of getting a big R from this individual, the probability of little r from this individual, that gives you 1/2, and the probability of getting a little r from this individual and a little r from this individual, that also gives you 1/2, which is what we did in the previous slide. Same thing over here, and the next thing that you want to do is multiply the probabilities that you get the expected ratios at both loci. So the probability of getting a big R, little r round and a big Y, little y yellow is going to be 1/2 times 1/2 because these are simultaneous events. So you do that for each of these possibilities, and that's how you end up with those expected ratios. you can carry that down to the F2 generation here and calculate those as well. So in this case, we've got, oh, this is a different cross. We've got a big R, this is a dihybrid cross. And let's say we're calculating just the phenotypic ratios. Up here, we were actually calculating genotypic ratios, which ended up being the same as the phenotypic, which is fine. So here we're just going to do phenotypic ratios just as an example. And we've got 3/4 expected from the R locus for a big R, for round, and 1/4 wrinkled. At the Y locus, we expect 3/4 to be yellow, 1/4 to be green. If we want to calculate how many we expect to be round and yellow, round and green, wrinkled and yellow, and wrinkled and green, we are calculating these individual probabilities for the individual crosses. So, whole different scenario. What are the expected phenotypic ratios for this cross? And this is a point where you can pause the video and try to figure it out for yourself. So again, the first thing you want to do is calculate the expected phenotypic ratios for the loci individually. And then you're going to calculate them together using the multiplication rule. Okay, so those are what we are expecting to get. If we were actually performing this experiment, if we actually did this cross, this is what we expect. What kinds of data are we actually expecting? So let's say we do the cross, we end up with a total of 542 seeds. How many then do we expect for each phenotype? Well, these fractions are also essentially probabilities. And we can use those probabilities, multiply them by the total, 542, and we get the expected number of seeds in each phenotypic class. So that's our expected data. What if we actually get something, not exactly that, but maybe kind of close? How do we know if our expected data how do we know that our observed data are close enough to our expected that we can conclude that our these traits are under Mendelian inheritance? All right, so in order to talk about this, we need to first talk about hypothesis testing, because that's what we're doing. We're testing a hypothesis. The scientific method, as we currently know and use it, involves hypothetico-deductive reasoning. And this is a method developed by philosopher Karl Popper in the 1970s. And what it means is that in order to do science, in order to use hypothetico-deductive reasoning, we formulate a falsifiable hypothesis that is testable, and then we test it. And typically, there are two types of hypotheses. There is our hypothesis, and then there's the null hypothesis. And the null hypothesis typically is often the opposite of our hypothesis. So, for example, it's important to note that in the media and in the way people often informally talk about science, we often say that something has been proven, that some principle has been proven, when according to hypothetico-deductive reasoning, nothing is actually proven. We can only disprove hypotheses. So we can only disprove or reject our null hypothesis, but we can't ever really accept our hypothesis. So if the data that we collect are significantly different from the expected numbers that we have calculated based on Mendelian principles, we are able to disprove our null hypothesis, and such that our hypothesis is actually supported. The data that we collect can only disprove our null hypothesis. This in turn supports our hypothesis, but it doesn't prove our hypothesis, because in the universe, we statisticians like to say that any result is possible, and we could have gotten a result that may appear to support our hypothesis strictly by chance. This chance of our results that falsely support our null hypothesis, that's a false positive, and that's a type 1 error. This is also known as falsely rejecting our null hypothesis. And calculating this probability is why we use statistics. The other type of error that we think about is type 2 error, and that's the probability of getting a false negative, that is falsely accepting our null hypothesis. So both types of errors become smaller when we have larger sample sizes, and this is why we like to have larger sample sizes when running experiments, because larger sample sizes give us greater statistical power. So many of you may have heard about a p-value that is associated with a certain statistic that can tell you whether or not it's significant. And the p-value is essentially the probability of a type 1 error, the probability of a false positive. The probability that we got the results we did just by chance and our hypothesis is not actually true. We generally use a p-value cutoff of 0.05. This is called our alpha value. By tradition, that was historically because of Ronald A. Fisher, who we've heard about. And we generally consider a p-value of less than 0.1 as what we call suggestive but non-significant. A p-value of 0.01 is strong evidence. 0.001 is very strong evidence, and 0.0001 is convincing. People don't necessarily strictly adhere to that terminology or that wording, but conceptually that is often how people consider and interpret p-values. So to put this in a graphical format, what you're looking at here is a probability distribution. And what you have on the x-axis are a range of possible observations. For example, number of seeds that are wrinkled. On the y-axis is the probability of seeing 5 here. This would be the probability of seeing, say, 5 wrinkled seeds in a given data set of 521, or however many we were talking about. And this distribution follows a normal shaped curve. And let's say if we had this observation, this was our data in black, and we have our cutoff of our acceptable p-value would be 0.05 over here. Because our observation falls within the range of what we would probably expect, then we can make conclusions based on that. This is essentially showing what I just said regarding interpreting different types of p-values, and that larger sample size increased your statistical power by decreasing your both types of error. So let's return to our experiment where our expected results that we calculated previously are shown here, and our actual results, the data that we collected, are here. So we want to know, are our actual results significantly different from what we expected? To do this, we use the chi-square test that we all know and love. And just to note, it is the chi-square test, not the chi-squared test. Don't square the statistic. I've covered that before. I have a whole supplementary video on the chi-square test. I highly suggest that you watch it if you are at all shaky on the chi-square. And even if you don't think you're shaky, you might learn some things from watching that video. So please take advantage of that. So this is a goodness of fit test. The goodness of fit tests are generally used with data that are counts of things. Number of eggs, number of pink flowers, number of scales. Those are all types of data that are appropriate for a goodness of fit test, such as the chi-square test, because they are counts and they are single integer data. All right, so our chi-square test tests if our observed results deviate significantly from what we expect. And again, they're appropriate for counts of different categories specifically. And so we calculate it using this formula that is very familiar to you, where we subtract the observed data from the expected data, or expected from observed. It does not matter because you are going to square the difference and divide that by the expected, sum it over the number of categories. So here we have 4 categories because we have 4 phenotypes: round yellow, round green, wrinkled yellow, and wrinkled green. And this is essentially how you would calculate it. Now, degrees of freedom. Degrees of freedom in this case are going to be the number of categories minus 1. In this case, it's when we use it to test Mendelian ratios and expected numbers, we use it as the number of categories minus 1. So in this case, it's 4 minus 1, or 3. Now, once you calculate this is our chi-square statistic. Don't square this. This just is what it is. That's the name of the statistic. And you go to your chi-square table. And what you want to do, how you read this table, there are different lines for different degrees of freedom. Ours is 3. So we go to this line. And we want to compare our chi-square value, 2.96, with the one that is required for a p-value of 0.05. This is our expected p-value, or the chi-square value for a given p-value for different degrees of freedom. And because our chi-squared value is 2.96, it's actually falling somewhere around here. Now this falls within the not significant range. What this means is that our results, are not significantly different from what we expected. So we can conclude that in fact, our trait does follow Hardy-Weinberg, that's a different lecture. Our trait does follow Mendelian rules of assortment and segregation. Okay, so that's basically all I had. I just want to say that testing Mendelian properties using a chi-square is a little bit different from the type of hypothesis testing that is usually done. Because in this case, we actually want a high p-value. We want our expected results to not be significantly different from our observed. In some cases, say you're testing drug efficiency, you do want, when you're comparing your treatment group to your control group, then you do want a significant p-value. But in this case, when we're using our chi-squared to test for Mendelian inheritance, we would like to be, our hypothesis is actually supported if we're in this range.